Welcome students. A brief knowledge and understanding of biophysical techniques is crucial in developing a better understanding of so many biological processes and diseases. And this further helps in finding out better diagnostic and treatment methods. One of the techniques that has propelled this knowledge is chromatography. In this lecture, we will have an overview of what chromatography is, what is its basic principle, what are the basic types of chromatography. We'll discuss the individual types of chromatographic techniques separately, and then we will conclude with the application of various chromatographic techniques. I must tell you, chromatography is a very vast topic and very deep topic. I here in this lecture only intend to present you with an idea so that it is easy for you to form a concept around this topic. So let's begin. The word chromatography has two roots to it, one chroma and other graphy. So the word chroma means color and graphy is writing. So the chromatography is the name of the technique which deals with the writings of the colors. So it's a method of separating physically two or more components which are distributed between two phases, the mobile and the stationary phase. It was in the first time documented by a botanist, Michael Sweat, who employed chromatography on plant pigments during his doctoral work. The term chromatography was coined in 1906, and it was not until 1930s that chromatography gained popularity and significance in the scientific work, and the importance that it gained later on can be identified because around 12 Nobel Prizes were awarded between 37 to 72 for the works which had chromatography in there as a very significant technique. So the basic principle of chromatography is the partition or the separation of the solute, which is also the molecule which we want to identify, between the two phases or the two solvents. So what is these two phases? The two phases or the two solvents phases we're talking about are classified as mobile and stationary phases. Now what is mobile? As the name suggests, it is the phase which moves. That is, it is either the liquid or a gas, and this phase carries the molecule or the mixture of molecules along with its movement across the stationary phase. And stationary phase provides the basic medium through which the mobile phase moves, and the sample also moves along with the mobile phase. So the interaction between the mobile phase and the stationary phase of the molecule which has to be separated forms the basis of chromatography. So there is a variation in how the molecule will interact with the mobile phase and the stationary phase, and this variation helps separate or identify the particular compound we are interested in. So these instructions are the ones which are based upon the physicochemical principles, and the physicochemical principles are adsorption, ion exchange, molecular sieving, or affinity. So these are the interactions which form the basis of chromatography. So what does chromatography basically do? With this technique, we're trying to identify either the composition of a mixture or to detect a particular otherwise known component in a given sample. The sample can be anything: a river water, serum, plant extract, a forensic sample, a urine sample for a dope test, etc. The sample is run through a stationary phase, and based upon the inherent characteristic property of that particular analyte, it is identified. Coming to the types of chromatography, the various types can be defined on the basis of what stationary phase is being used and what mobile phase is being used. So when the stationary phase is solid and mobile phase is liquid, the chromatographic method is either column chromatography, thin layer chromatography, ion exchange chromatography, or high performance liquid chromatography. Whereas when the stationary as well as the mobile phase is liquid, the chromatography type of chromatography becomes column, thin layer, HPLC, and paper chromatography. And when the stationary phase is liquid, whereas the mobile phase is gas, the type of chromatography is gas-liquid chromatography. So coming to thin layer chromatography, it is one of the simplest forms of chromatography, just more complex than paper chromatography. In this chromatography, a thin layer of solid adsorbent, usually formed by silica or the alumina, which forms the stationary phase, is coated or layered on the sheet of maybe glass or metal or plastic. So what happens, this after this silica or the alumina is coated on whatever sheet we want to use, it is dried and a small amount of mixture is applied at the bottom, and this plate is then placed in a solvent chamber which is called chromatographic chamber, and the as the liquid moves up via the capillary action, the mobile phase moves, which is formed by the solvent, and across the path of the mobile phase, there is a separation of various components of the sample applied. So it is based upon the differences on when a particular out of the mixture of the components at what stage or when a particular component is adsorbed and at what stage it remains in the solution. So this figure should help you understand whatever I just mumbled. On the extreme left of the figure, you can see a rectangular gray strip. This represents the chromatographic sheet. Below arrow indicates the increasing development time of the chromatographic sheet, and this right hand arrow indicates the direction of the movement of the solvent. So as you can see here, this is the spot or the place where we put the sample. So this is the place where the mixture will originate from. This below line indicates the level of the solvent. The sheet will be dipped only up to this amount of solvent in the chromatographic chamber. It will not be more than that. So this forms the solvent front. When the sheet is placed in the chromatographic chamber with the solvent and covered from upside from the top, the solvent starts moving with time via the capillary action. It crosses the origin and the individual components start separating. You can see first component A separates, then component B separates, and this arrow indicates the solvent front. With further increased time, the component B moves still further. So In thin layer chromatography, the separation of the compounds is based on the competition of the solute and the mobile phase to the binding sites on the stationary phase. So this is a very simple and easy representation of thin layer chromatography. Now that was about the performance of thin layer chromatography, but how to analyze it, how to draw information from that. So an important factor we need to understand here is retention factor or RF factor. The retention factor is an indicator of how far a particular compound travels in a particular solvent. We should know that it is a characteristic feature of every particular compound. So whenever we develop a chromatographic plate of a particular compound using the same solvent and same stationary phase, same retention factor will be achieved. So it's like a signature of that particular component. So thus this plays a crucial role in comparing the new compounds with the compounds, unknown compounds with the compounds of known retention factor, and hence it helps in identification, placing the identification of those new or unknown compounds. So how is it calculated? It is calculated as the ratio of distance moved by the solute to the distance moved by the solvent. Now coming to gel filtration or gel permeation chromatography, this technique is based on the difference in the size of the solute molecules which are to be identified. So the sample that which is an analyte for us, has a mixture of components, and all the molecules which are present in that mixture have different sizes. So, and once these are applied, these move through the stationary phase, which is the beads, which are porous, and so according to the size, whether they take longer or smaller to pass through the stationary phase decides which molecules will be eluted at what stage. Let's just try to get a hold of what I just said. So first of all, the gel permeation or gel filtration chromatography is conducted in a column, unlike the thin layer chromatography where sheets are used. So these glass columns constitute have the resins or beads which are porous, which are shown as gray in here. So these beads are packed, solidly packed in the columns. So what happens is, you can see the beads have various sizes, and so once the sample is loaded onto the column, at the top of the column, uh, the different molecules in sizes are represented by red ones and the blue ones. So the red ones are the bigger molecules. They are unable to enter the beads as their size is larger than the pore size of the beads. So they just move along with the mobile phase, but without entering the porous beads, and they just trickle down. Whereas the smaller molecules, which are indicated in blue circles, tend to enter the beads because of their smaller size, and their pore size is their size is comparable to the pore size of the beads. So as they move through the pores of the beads, they take longer in moving along with the mobile phase. So what this longer and shorter time means, the larger molecules will be eluted first, they will come earlier, and the smaller molecules will be eluted last. So what is done? So in the right side you can see of the figure, you can see the same process is depicted, but this diagram clearly shows that as the the larger molecules are eluted first. So what is done is that uh, these eluent or the mobile phase is collected in small volumes called aliquots in separate vials, small vials, and so it is expected that one vial should contain one type of molecule. Like you can see, all the red ones are collected in one vial, and the rest next vials will contain the green ones. And for simplicity, only two components are shown here, but of course there can be a higher number of components in the mixture. So basically, this is about the components being filtered through the resin molecules. Gel filtration finds its most common application at determining the molecular mass of a protein as an unknown sample is run against a standard solution of known sizes. So next is affinity chromatography. It is based on the property of binding capacity or the affinity of a particular molecule to the stationary phase. So the interactions involved in the affinity chromatography are as specific as between antigen-antibody interactions or the enzyme-substrate interactions. So in the process, uh, you can see, let's first understand the key. So the triangular is representing the target protein that we want to separate, while all the other figures represent the other components of the mixture. This red small circle represents the salts. This greater than sign, smaller than sign shows the indicates the ligand, and this is the affinity resin or the beads or the matrix which constitutes the stationary phase of the affinity column for this affinity chromatography. So at the first step, we have the beads or the resin loaded in the column which has the specific ligand bound to it, and all of the sample mixture which has all the components including the target component or target protein is loaded onto the column. Based upon the specific binding, only the target protein binds to the ligand, and rest all of the molecules along with the mobile phase elute, move down, move through the column and are eluted out. And when we want to actually elute the uh, protein, the a high salt concentration or a different pH condition is pH solution is used. It destabilizes the binding of the protein to its ligand, and eventually when there is only one protein in the column, that one protein trickles down with the eluent or with the mobile phase and it is collected. The the best feature of affinity chromatography is that it is capable of providing highly purified fractions of the required target protein. So now let's come to ion exchange chromatography. Now this chromatography is based on the binding of opposite ions and repulsion of the like ions, and also the relative higher or lower positive or negative charge of a particular component. So let's just simply understand, a column having beads is packed. So the beads in this column carry the charge either negative or positive. In this case, in this example, the beads carry negative charge. Column is packed. Sample containing the mixture of proteins is loaded onto the column, and then it is eluted with the mobile phase. So with the movement of the mobile phase, the proteins will also move along with the mobile phase, and the rate at which they are eluted from the column is determined by their net charge at the pH being used. Now, ion exchange chromatography uses two types of resin beads which constitute the stationary phase. One, cation exchanger, and another anion exchanger. A cation exchanger has negative charged beads which will attract the positive ions, whereas an anion exchanger will have positive charged beads which will bind the negative ions. So, like in this example, which is of cation exchanger, the positive ions remain bound to the negative beads, whereas the negative ions flow through the column unbound. So, let's get to gas chromatography. Uh, it is the method of separation of volatile components and is based upon the partition of the components between the mobile phase and the stationary phase, the only difference being the mobile phase is the gas phase and the stationary phase can be formed by either liquid or by solid. So the high level of automation is incorporated in the gas chromatography. The components which are separated as the gas moves are detected by a detector. The gas liquid chromatography variant of gas chromatography is highly sensitive, rapid, and is very reliable. It's frequently used in the estimations of various biological materials such as lipids or drugs and vitamins. Now, this diagram helps form a basic understanding of a gas chromatograph. The carrier gas, which is usually an inert gas such as helium, is mixed with the sample through an injector port, and both of these are moved passed through the long gas chromatography columns which carry the stationary phase. The eluent coming out of the column is detected by the detector, and the detected messages are analyzed by a computer operated software. So next is a very important chromatographic technique, high performance liquid chromatography. It's a very powerful technique. It has a stationary phase, which is an immobilized thin layer of liquid on the microglass or the plastic beads. The mobile phase is constituted by a solvent system, which is passed under high pressure through the column. The conventional methods of HPLC are 3 to 25 centimeters long with very small internal diameters as small as 4.6 mm. So you can imagine that in order to pass a liquid phase, a mobile phase, through this thin a column, high pressure will be required. So the pressures of as high as up to 50 megapascals need to be applied to the columns, and since this high level of pressures are applied, this makes this chromatographic technique a high performance chromatography. And just like the gas chromatography, this diagram also helps us understand the high performance liquid chromatography working. It's also as simple as that. There is an HPLC solvent or high performance liquid chromatography solvent, which is via the HPLC pump injected into the injector, and the sample is mixed with the solvent. Both of these are injected into the HPLC column containing the stationary phase, and the eluent is further detected by the detector and analyzed by the requisite softwares. So due to its rapidity in action and its performance capability, The HPLC is used in assaying multitude of components. These are amino acids, proteins, carbohydrates, lipids, nucleic acids and related compounds, vitamins, hormones, metabolites, and drugs such as antiarrhythmics, antibodies, antiepileptics, analgesics, bronchial smooth muscle relaxants, and antidepressants. So you can see the wide applicability of HPLC. After this brief description of these various widely used chromatographic techniques, let's just discuss the application of these chromatographic techniques. So liquid chromatography is widely used in testing water samples in order to gauge the water pollution. Gas chromatography is usually used in forensics labs uh, and to detect bombs in the airports and in order to identify drugs or alcohol. And thin layer chromatography is used in identifying pesticides in the food or the dye to identify the dye composition of the fibers, again in the forensics laboratory. Paper chromatography is used in RNA fingerprinting and in separating components such as anions or amino acids or histamines and antibiotics. So these are the sources that I have used while preparing this lecture. So if you've reached here, you must be very patient listeners. So I thank you for listening it until here. I hope I was able to give you a hint of what chromatography is and what are the basic types of chromatographic techniques. So with this, I thank you for being a patient audience.